morning. I'm Milind Gore. To start with, I thank IMAPAC and Vaccine World Asia Congress management people. What I'm talking about is APC promoter-based DNA vaccine. Now, because we are in a COVID era, now just a brief on the COVID. That one of the problem is we get a differential symptoms of the asymptomatic to mortality and long-term and mortality, etc. are there. Severe cases of COVID, they exist, has of course death is also there, but it also has the post-vascular and coagulation and neurological also are there. So one of the thing is, as an immunologist, one of the thing is antibody response against the COVID is only for the N protein spike also, in addition to other proteins also, N, E, and M also. One of the biggest problem currently is the spike protein immune response remains very short term. But the other proteins which are there, N and E and M, it remains for more than 1 year. And simultaneously, in addition to T cell, B cells, there is a T cell response also is there. And what has been seen by the recovered patients and etc. So they have seen the T cell responses against the E and M and N and not very low for the spike. That is one of the problem of the current vaccinations which are giving. And protection is also there. If the people have the some kind of other proteins, antibodies and immune response is there, then the protection is higher protection. And that is what is happening in those things. And other things are routine, standard things. Now, to brief on the processing, how the immune response is processed in that, everybody knows that. What we're focusing is co-stimulator is important for the developing the long-term immune response, and that has to be focused on the everywhere. In the absence of co-stimulator antigen, in the BSL, BCR, TCR kind of thing, then if it is not there, it will lead to low affinity maturation, immune response, especially antibody. And the low affinity will lead to anti-self immune response. And that is what is the biggest problem in the current situation. And that's why we are getting the other things are there. It is possible to bypass the co-stimulator in the beginning, but it should not be protected quickly, and then only we'll have long-term immune response. Now, for all this, monocyte-based APC are there, antigen presenting cells. They have the co-stimulator and the things are also there, and that's why these things are essential, and which generate the long-term immune response, and course things are there. And antigen presenting co-stimulator result in it removes if it is not there. It will not, if it is not there, it will result in anergy and autoimmunity also. Well, that is one of the problem, because in a current situation, overall years now, 4 dose also antibody response is lower, bringing down, and that is leading to the variants are coming up. Now the intermuscular and intradermal also are there now looked into, and everybody understood that T cell response is required. So that is a very recent thing is coming up, and it requires the pre-existing antibody. What happens is, we all have the common cold in the childhood, and that T cell epitopes of E, especially N protein, these are there as the memory cells in our body. And if you can use that as in a vaccine, it will help the long-term immunity. And now the thing is, we are talking about anyhow we are focusing on the vaccine. So what happens is, why the DNA vaccine? To begin with, currently mRNA is protecting the people, but it may have the problem of other temperature, etc. are there. And one of the advantage is what happens is, because we are injecting in intermuscular, it enters into non-immune response also. But because the cells which are myocytes and other things, fibroblasts, etc., which are muscular, they will be insertion of the mRNA will go, and they do not have the co-stimulator, and they develop the whatever we were talking earlier. And what happens is, because of that, currently 2 year study by FDA was there, and they saw that the mortality is there in a, and escaping the other variants are coming up. And the major problem the FDA studied is myocarditis and pericardiac patients are doubled in the USA now. That is what is the problem of COVID now. And in terms of vaccine, now there is only one study now that there is mRNA which combines both an S and N protein mRNA. That has been studied. Now it has been studied only at the animal model only. Now the advantage of course, is temperature, etc. Stability is there, but disadvantage is the is essential to enter into nucleus, and that is one of the other problem of the DNA vaccine. And currently DNA vaccine which have the CMV promoter, they are not approved for the animals, but it is not for the humans, and that is why the problem is there. Now, as a rule, antigen or plasmid or everything goes into the cells. It enters into the nucleus, especially if it is the DNA and kind of size, etc., etc. So that has to be seen into, and that will happen. That is what we are focusing on that. And the current DNA vaccine, again, I'm specifying, the CMV has a problem that we have the high level of more than 80% are positive. And to overcome that, people have tried to use the IL-17 also, and it can be used. And other earlier we have the CMV immune response is there, so that also is there. And that is why the currently CMV vaccine is used for animals and it is not for humans. Now, the thing is, in 2005, I started doing it. What happens is, our studies, we started that instead of CMV promoter, we started thought about the APC promoter. So in a mouse model, we used a long term back in 2005 to 2011. So we chose the proteins which are specific for monocytes only. And we chose the 3 proteins, and the promoters were used of those. Macrocillin, EMR, and beta integrin was there. And we use the monocyte, we use the raw cells. And for fibroblasts, we use L929. Now, once you see this, CMV promoter and etc., everything was studied simultaneously. And the CMV promoter enters into the all cells, whether it is a raw and whether it is L929, and it remains very long term. And because this is what is the problem of non-co-stimulator cells. So that was what is happening in CMV. And in addition to the 3 protein promoters, the macrocillin was the best because it remains for the long-term thing, because GFP was the model we used for the study in vitro. And other promoters did not work very good. So because it was better than others, so we chose macrocillin for the future study on Japanese encephalitis. And one of the thing is, absence of the expression of GFP was not because of it was not entered, but the level of transcript was similar to the any other, both the cells were same. That is what, so that has been presentation is also there. Now, the thing is, we use the whole thing, and the plasmid was there. So we replaced the JP with the E protein, and we studied it, routine methodology of encephalitis kind of thing. We had a monoclonal antibody, etc. And what happens is, ELISA, we got more or less equivalent ELISA with CMV and other thing, a microserine also. And neutralizing antibody is also more or less same. And we challenged it with the challenge the immunized mice, and the production was more or less similar. 87 in CMV and 76 in E protein of J, which is kind of gave us a chance to decree, have a confidence that it can work. Now, what happens is, now that the whole study was there for the only for the this thing for the animal model. So now the thing is, we started looking at the human similar kind of thing. So eventually I found that macrosaline, which is in humans, it is called as CD68, and which is presented only in monocyte and Langerhans cells only. And other cells have a slightly different things. And that's why we have to use macrosaline or CD68. However, one of the thing is, CD68 should be modified by in vitro, in silico, that it can be used for the mouse and primates and other animals, and that can be done. It is a basic bioinfo kind of thing, and this is what can be done. And if you insert in the same plasmid, bisyndromic vectors are there. So what will happen is, if the something different comes up, we can add the B cell epitope, T cell epitope, chimeric, other things which have been studied earlier by me in my JE things. So that can be used, and it will be used for the influenza also, in addition to the COVID. And to overcome some of the current problem of T cell epitopes, we have developed a de novo modified protein also is there with us, which is under publications now. And let us see when we come in. The 48 is phrase is there. Now, to overcome, how to overcome the gene gun kind of thing, which is another cost basis and low economy income countries. We have to have something which is low cost is there. So nanoparticle has been used. That is where the nanoparticle comes in. And one of the thing is, nanoparticle is being used for quite some time. Even in humans also, it approved, and cancer, autoimmunity, etc. they are used. But these are biodegradable polymetric micro nanoparticles are there. And there are many nanoparticles are there. And Catosan is one of the seems to be best, because it is but the temperature making the nanoparticle with catosine has a temperature problem is there. So DNA will overcome that problem. And initially there was a problem that it was a neutralizing kind of thing. It did not have a charge. So studies are already been done, and the positive charge has been inserted into the catosan, and that is what is giving the good time. And what happens is, the catosan, when it enters into the cell, it enters into cells, it will enter into nucleus also. So without the gene gun, catosan promoter nanoparticle can be, we can encapsulate the APC promoter plasmid into catosan nanoparticle thing, and rest of the thing is purification of plasmid. And what will happen is, eventually, for whatever the antibody, whether it is COVID or other things also, one can use it for the just inoculate, give the administration of that modified plasmid thing, which encapsulated with catosan, just by intramuscular. And our other advantage of we can insert the T cell epitopes in the gene kind of thing, and that can be used. And suppose there is a new promoters or new variants are coming up, despite of that, that will be that can be converted quickly into the thing. One of the advantages now, all the vaccines which are giving is below above the 15 area. Now, although children are in age is increasing, so we have to focus on the children now. How to immunize the children as early as possible to protect them from the COVID in future. If you if internationally, if you can do it, then the COVID will be come down to the zero kind of thing, and that is the aim of what we are talking about. And that is the thing which I feel which can be taken up. And of course, I do not have any lab, and so I need a lab support from somewhere, and that is what is there. Now, I must acknowledge the Dr. Firaz Ahsan, who did all the work, lab work, he did his PhD under this. And of course, Charles Darwin said that intelligence is also there in addition to the variants also which can be done. But we are humans and we have intelligence, and that's why we'll survive the and other infections also in long term. So thank you and take care and be safe. Thank you.